I'm the head of outreach at Protocols.io, and today I'm going to share with you this webinar about reproducibility, the methods behind the data. And our agenda for today is, first, I'm going to show you what the motivation behind Protocols.io is, and then I'm going to tell you what Protocols.io is all about and the main functionality on the platform. And next, I'm going to show you an example group, how people are collaborating, and it's the Mouse Metabolic Phenotyping Centers group. And then I have a little fun exercise for you and a section of the art of writing protocols, because there's really an art behind writing research protocols. And then in the end, we have some time for questions. And so I would like to start with this article. And so this article here, it's from a project that started about, I think, 5 years ago. And it was a plan of researchers. It was a really, really high, how do you say that? It was like a plan to replicate 50 high-impact cancer papers. And so scientists were really motivated to do this, and everybody got very excited to replicate all these very important research papers. And so scientists got together, and after just a couple of years, the project shrank to trying to reproduce 18. So from 50 papers that they wanted to reproduce, they decided to just have 18 in the end. And the main reasons for why they started to stop at that point and they didn't try to further reproduce all the other ones is that most of the ones that they tried to reproduce, it took a very, very long time. It took much longer than expected. And here are some reasons why they stopped and the lessons learned from that project. So here, for example, Fox Media said scientists and journals need to get better at describing their methods and sharing data. Or here, like in many cases, because the protocols had been developed by students and postdocs who were no longer in the lab. So it was really hard to figure out what actually was done, and people could not find the original people that executed the protocol, executed the projects, and so it's really hard to track down. And like here in this one, it says the hardest part by far was figuring out exactly what the original labs actually did. Scientific papers come with method sections that theoretically ought to provide recipes for doing the same experiments, but often those recipes are incomplete, missing important steps, details, or ingredients. In some cases, the recipes aren't described at all. Researchers simply cite an earlier study that used a similar technique. And so this is really a common issue that we have in science today. And this is another tweet that kind of describes something really well too. Like here's a scientist, he was looking for a protocol in a 97 paper. That paper said that the methods were done as described in a 96 paper. The researcher finds the 96 paper that says, oh, we just did this as described in the 87 paper. Then he finds the 87 paper, and then there's a paywall and he cannot access it. So he doesn't know what was actually done. So that's not a really pleasant experience. Or here on the right-hand side is another tweet from a physicist. He was reading a paper from 2017 that said devices were fabricated as previously described. He finds that reference that says devices were fabricated as previously described. He finds another reference, previously described, and then the very original paper says devices were fabricated with conventional methods. So again, he does not know how to fabricate his devices now. And so the mission of Protocols.io is really to make it easy to share method details before, during, and after publication. So that's what we're trying to do with Protocols.io. We want there to no longer be any of these issues that I showed you before, because we really want to make it easy for everybody to just share all their method details and all the details that are needed to make a method reproducible. And how Protocols.io works, the main functionalities are really discovering. You can discover publications. It's an open access platform, so the public content is free for everybody to read and access. You do not even need an account to discover all the publications. Then you can create your own protocols. You can share the protocols with others. You can publish the protocols, of course, and you can collaborate with others. And then there's something called running protocols. So that is really if you're in the lab doing the experiment, we have something called a running feature, and we also have that on an app. So you can basically check off the steps as you follow them. And then last, I'm going to show you the file manager. So first, for discovering publications, when you come to the landing page on Protocols.io, you will see a little search window up here. I put a pink arrow. And this is where you would type out your keyword that would be relevant for the protocol that you're looking for. And you just type the keyword and hit enter, or you click the little search icon. And then once you do that, you will get a whole list of all the protocols that might be relevant for that keyword. And you can sort by different things such as views or activity or publishing date and stuff like that. And you can also search for example, for other researchers or groups or reagents and materials and stuff like that. But then when you look for the publications and you find a protocol that looks good to you and you want to look at it in closer detail, you can click on it. And then this is how a protocol page looks like usually. And the special thing to note here is that those protocols are not static PDFs, but they're rather very interactive and dynamic protocols. So there are a lot of things happening here, and I'm going to walk you through what the main things that you can see on this protocol here. So one thing is protocols can be associated with a group. So here, this is an example from a group from the Human Cell Atlas Method Development Community Group. And next here are steps. So the protocol is not really one big blob of just text, but it's single steps that are broken down in very easy, like just one task per step things, so people can really easily follow the method as they're doing it. And then on the right-hand side here, you can see a commenting functionality, and this is really to discuss the protocol. So you have this commenting functionality on the public protocols, but also on a private protocol. So if you're having a protocol that's private and you only want to share it with your internal collaborators or like just with close collaborators, you could have that same functionality on a private protocol as well. And so how this works is really there's two different levels of how a protocol can be discussed. And it's either on a protocol level, that means people can ask questions, make suggestions, or improvements on the whole protocol in general, or if they have a question on a specific step, there could be a discussion happening also on a step level. So if, for example, you find a protocol and you have a question about step 2, you could click on step 2 and add a comment to step 2, and you could say, for example, why did you do half an hour incubation and not an hour, or whatever that might be. And then everybody who's following this protocol and the author, of course, will get a notification once there's a new comment, so they know that there's a comment, and then usually the author or somebody from the network will respond and try to figure out whatever the question was. And then, of course, the protocols can be linked to publications. So if your protocol is part of a publication, it can be linked to that publication. And then also the publication will link to the protocol. So it's like the protocol links to the publication and the publication links to the protocol, because every public protocol receives a DOI, which is the digital object identifier. This is really a link that will be permanently there. It will always link to this protocol and it will never change. So this is really the link you would include in a publication. And then here, one more thing that's very special about Protocols.io is we have an option for versioning. And what that really means is that you can keep the protocol up to date even after it was published. So sometimes you might publish something and then usually you keep working on that same method and you keep improving it, especially as there's discussion happening. Maybe there's some things that you'd like to improve. So you can always create new versions of the protocol and make adjustments to keep it up to date. And so if somebody, for example, has the GUI of an older version and clicks on that, and there's a new version available of the protocol, they will get a little pop-up that lets them know that there's a new version available. And then people can decide if they want to view the newest version of the protocol or the older version. And then there's also an option to compare the both. So there's a button that says compare, and then you can click compare and you can compare different versions. So if there's 5 versions, you can compare version 5 and version 1, or version 2 and version 4, like whichever version you want to compare. And really here it just gets highlighted what were the changes and what had been modified. And then next is the running functionality, but I'm going to tell you more about that after. I'm going to show you how that looks like. And then if you would not like to run the protocol through our running functionality, of course, there's an option to export the protocol, so you can print it out, or you can save the PDF to either directly your computer or Dropbox, Google Drive, or Box. And then you can have a paper printout for the bench if you're doing the protocol, if you're following the protocol in the lab. And so Protocols.io is, as mentioned, is an open access platform. That means it's really free for everybody to just access all the public content. There's about 4,000 public protocols right now. And also, one thing I would like to mention is that there's a large diversity of disciplines. So it's not all just molecular biology protocols, but we also do have psychology protocols, computational workflows. There's a lot coming up, and clinical trials and teaching, just to name a few. If you're interested in seeing different categories as well, feel free to explore more on the link I provided down here. It's just protocols.io/explore. And then if you are coming to Protocols.io and you would like to create your own protocols, you can do that really easily with one click. You can click on the little plus button, which is up here next to your profile, and you just click new protocol, and this will lead you to our editor. And so there's really a lot of things you can add here. You can add guidelines and warnings, a description of the protocol, materials, and then the individual steps. And so for the steps, you can really add a lot of images and amounts of stuff. You have all these components to work with. So if there's like an incubation time, you can add the timer to it directly already, and all these things. And there's a closer look up here. So you can divide the protocol into different sections. So you can group it out into sections, and then you can add the exact amounts. This is good for later if you, for some reason, want to scale the protocol or some steps of the protocol. If you use those components, you can, for example, say, I want to scale this step by 3, and then here, instead of 300 microliters, it would tell you to add 900 microliters. So this is really convenient. And then the timers, of course, if you run the protocol, it will start automatically if you click start. So you don't need to set a timer for anything individually anymore. And then, yeah, you can really add a lot of detail to those steps. And also, yeah, here on the right-hand side are the components. And then if you add videos, you can add it through this. And then also, what we really encourage people to do is to add very detailed materials. So you can either, we have a lot of materials already in our database, so you can search for available materials by either the material, the reagent name, or the catalog number, or the vendor. But if there's a material that you're using and it's not in the database yet, you can add a new reagent, and you can really add a lot of details here. So you can put the reagent name, you put the vendor where you got it from, the catalog number. You can put a store URL that goes directly to the vendor where people can buy that protocol. And then you can put a CAS number if it has any. And you can, of course, you can put an RRID to it as well. And that will go directly to the RRID portal. And Yeah, so really for the protocols of protocols.io, you can add as much detail as you need, and there's not really a limit of what you can add. And then also, as mentioned before, it's not only wet lab stuff, but if you have computational workflows, there's also an option to add all the computational components, so you can put all your data sets and code and all these things here for computational methods. And then once you created a protocol and you're ready to share the protocol, there's different options. So every time you start a new protocol, it will always start out being private to only you. So only you're the only person who can see the protocol. So it's really up to you who you want to share it with, if you want to make it public or anything, but it always starts out private. And so if you want to share a protocol privately with others, you can either do that by getting a shareable private link. You can do that by clicking a little link icon here that will open up in the Share with Others screen when you click the share button. And that allows you to share the protocol with anybody, even if they don't have an account on protocols.io yet, because with this link, they can access the protocol. Just please be aware that if you share it with that functionality, people will only be able to view the protocol, but they cannot make any edits or changes to it. And if you want to share it with somebody who does have an account on protocols.io, you can share it either with individuals by typing their email addresses here or by searching for their names here. And you can also share it with an entire group. So if you're part of a group that has a group set up on protocols.io, you can just add that group, and then automatically it will be shared with everybody in that group, and they can access it. And then you have the option to decide between different sharing settings. So you can decide, do you want that person or that group to be able to only view the protocol? Should you be able to also edit it? Do you want them to have permission to edit and publish it? Or you can also make other people a co-admin of this protocol. And then if you created a protocol and you're ready to make it public, you will see two different orange buttons on protocols.io on your protocol. One is called Get UI and one is called Publish. And so the main difference between those two is really that the Get UI button is more for if you are submitting a paper and you would like to still kind of keep your protocol private, so you don't want people to find it on protocols.io yet. So if you click DUI, Get DUI, it will give you a DUI for your protocol. It will be permanent. You can no longer make any edits to it, but you will have the DUI to include in your paper that you're submitting so the reviewers can access it. So everybody who has the DUI link will be able to access the protocol. But if somebody tries to search for that protocol in our search functionality in Google or any other search engine, it will not come up yet. So it will be still kind of private, but it will be having a DUI, so you can share the DUI. And if you're ready to make it fully public and discoverable, you can use the option called Publish. And this will also give your protocol a DUI, and it will be permanent. You can no longer edit it, but it will also be showing up in the search results. And yeah, if somebody searches for it, they will be able to find it. So those are the two different options you have just to keep in mind when you're publishing a protocol. And then if you're publishing a protocol and you would like to associate it with a group, you can, of course, associate it with a group by here, like when you're publishing, we ask you to follow these steps here. So you put a title, you set the authors of the protocol, and you set a status also. So for example, we ask you, is this protocol something that's working? Or maybe it's a protocol that's in development, or maybe even it's a protocol that you tried and it didn't work. So you can really put what the status of this method is here. And then you also have the option to add a group. So for example, this is an example for a human cell atlas group. If you switch these on, it will the protocol will have a link to that group if that's a public group. And then once a protocol is public, it is citable, of course. So every protocol gets a citation that people can cite. and add to their manuscripts if they're using that methods. And also every public protocol is archived in clocks, which ensures long-term preservation of the methods. And people can add it to public groups to organize and discuss the methods. And there's an option to for everybody to fork that method. That means they can create their own version of your protocol of it and just modify it as they need it. And you, of course, always have the option to create a new version as needed. And so the main collaboration functionalities and protocols are the groups. So I mentioned it before, but you can create a group that can either be private or public, and you can have all your collaborators in it, or it can be a broader scientific community that collaborates on a different field, on like a field. And then every protocol has the commenting functionality and versioning and forking to keep the protocols up to date. And easy sharing of protocols, of course, and experiment records and files with each other. So you can share all your experiment records also through the file manager in the groups, or like it has the same functionality as I showed you for the protocols. You can do the same for the experiment records. And then running protocols. So if you have a method, either a public method or as if it's a private method that you're following for your experiment in the lab, you can get the running functionality going. And so you can get basically a step-by-step description of the protocol, and you will have this checklist that you can check off every step as you complete the step. And so you can either do that on a computer, or you can also do that on a tablet or phone. So we have free Android and iOS apps that you can download, and you can just have the same thing that you see on the left here on a phone, that you can just check off the steps. And then every step that you check off, of course, gets a timestamp. That makes it really easy to just track back if ever you need to go back to see what you did on a specific day, because everything gets the timestamp with the day and the exact minute and second. And then if there's anything for some reason you do differently on one day, you always have the option to edit the steps. So, and that if you edit a step here, that will not make any changes to the original protocol, because this is just your experiment run. So you can make a change and say, today I did a 2-minute incubation instead of 5 minutes, or whatever it might be. And you can also add any notes and pictures. So that's really good for observations, to really just make it clear what you did on that specific date. And then next is the file manager. So the file manager really is your space to keep all your work organized. So all private protocols that you start go into here. And then all your group files you can also find here. So every group has a group folder that's really one space that every group member can access. And so this is really your space to organize everything, and you can move files around by just dragging and dropping them. And you can also add different kind of files in here. So if you have any spreadsheets or PDFs or Word documents that you would like to put into your file manager, either in your private files or in a group folder, you can add them here as well. So it's not only for protocols, but really for any kind of files that you would like to add here. And then this is an example of a public group. It's from, some of you might know about this one. It's the Mouse Metabolic Phenotyping Center's protocols group. And it can be found on this link that I provided down here. And really in this group, you can see, let's see if there's a video. It should be a video. So here in the group, you can see a timeline that really shows you all the things that are happening. So if there's a new protocol posted, you will see it on the top here. And then there's a little description of what this group is all about. And then this group has 218 publications now, so you can really browse all the publications of this group here, and you can sort them by date or activity name and views. And of course, you can also search within this group. So if there's anything you would like to search for, you can search. And then every of the protocol cards, every card has like indications of how many views does it have. And if a protocol has a comment, it will also show comments here, and also forks and different indicators like that. Let's see. Oh, sorry about that. And one thing I would like to show you what could happen if so many protocols are in an open access repository like protocols.io is that here's a great example. So somebody tweeted that they were looking for someone with experience in doing RNA extraction from primary cortical neuron cultures. So, and then there was like a discussion happening here, and then in the end, people suggested that they should try this protocol, which is an RNA extraction protocol for trisol protocol. And the cool thing about this is that if you look at which paper this protocol is part of, it's a paper for stickleback parasite. So really, it's like very unrelated, like because this person was looking for something for an RNA extraction from primary cortical neuron cultures, and then somebody suggested this, and the original paper comes from a stickleback parasite paper. And so it's really cool because that person would have probably never thought of looking for a method in a stickleback parasite paper. And so it's really exciting to see that those kind of things can happen, that the methods could work even though it's like an unrelated field, because you really have all the different methods in one place, and people don't need to go through all the papers to try to find a method that might work, because all the methods are right there. And so that's something very exciting that can happen if you have a large protocols repository such as protocols.io. And then before we get into the next part, which is the art of writing protocols, if you guys have a pen and paper close by, that would be great if you could grab that, because I'm going to do a little exercise with you. So I'm going to give you a minute to just grab a pen and paper. It doesn't need to be anything fancy. It can be a little paper and just a pen. And so what I would like you to do now with your pen and paper is I created a protocol here. It's a drawing protocol. And what I would like you to do in the next 5 minutes is to try to follow each of the steps from starting with step 1 and just draw what is described here. And then I'll be very excited to see how that goes. So I'm going to look at my time now, and I'll give you about 3 to 5 minutes to do this. Maybe 5 minutes. And if you have any questions, please feel free to unmute yourself to ask questions or type them in the chat window. Okay, so if you drew it, you might have something that looks either similar like this, or maybe it looks like something else. And so this is from, I did this exercise with another group at some point, and these are the results from that group. And so the interesting thing here to note is that even though everybody has the same instructions, you get very different results. So so it's one protocol, but many results. And the description that I wrote for that protocol was really was this. So it was SpongeBob, but not everybody looks like exactly the one that I had in mind when I was making those descriptions. And really what I want you to note with this is that the description, of course, my description was lacking some detail and it was not accurate enough. But we have that problem in science a lot. Like here's this person, he posted, he wrote an article about, he said that, so when he's reading the method paper, sometimes he feels like the method section, as if you would describe it, how to draw an owl, it would read like this. We draw the owl on a 60.2 GMS white paper of the A4 dimension using 3H and 6B graphite pencils. We did so by looking at owls and drawing what we saw on paper. This protocol yielded one drawn owl. And so, of course, here there's a lot of, this is very exaggerated, but really in papers we see that a lot happening, that there's really a lack of the details and you don't really know what was done. And so some tips that I would like to give you to sum this up is, if you are writing protocols, it's always good to digitize them and add a lot of details to your protocols. So I encourage everybody to use a digital way to write your protocols. So for example, protocols.io, or you can even do Google Docs or any electronic lab notebook. But if you write it on paper, it will make it really hard mostly for yourself, because if you, for example, in a year, you would like to try to track back what you did on a specific date, it's really hard. But also if you want to share it with anybody, it can be very difficult if you have it written on paper, because the paper is not searchable and it's very hard to share with others. So it's really good to have it somewhere digital. And if you're writing a protocol, think of a protocol as a brief, modular, and self-contained scientific publication, because it really is. It's like really a publication in itself almost, because it has so much details. And also, if you're writing a protocol, it doesn't matter where it is, but it's always good to include a 3 to 4 sentence small abstract that puts your method in context, because that method might be used by somebody many years later, and that person might not know what the context of your protocol was or what you were working on when you used this method. So it's really good to just put a little abstract in front of each method to just let the reader know what this protocol is all about and the context of how you were using it. And then, of course, again, include as much detail as possible. So add all the reagents you use with all the exact details to it. And also you can add different things like the amounts, and really put a lot of detail. So if you're shaking something, don't just say shake for 5 minutes, but tell people, how did you shake it? Did you shake it by hand? Did you shake it with a shaker that rotates? Like, what kind of shaker were you using and what temperature? And like, really, it's the little details that matter and that make a method be able to reproducible or not, because really you need all these little details to be able to replicate a method. And then also very important is the chronology of steps. So if you have a method, try to not mess up the chronological order. So if you say, for example, I did this and this prior to this and this, and like it's really important to keep the steps in chronological order. And then also, I can encourage you to limit jargon and use active voice. So it's really better to use an active voice and not use anything like past tense, or just say, oh, the tubes were shaken and stuff like that. Like, say, shake the tubes. So really use active voice, and try to not have any access like jargon that might just be more confusing than clear, because always methods, they should be understandable internationally, because you never know where that method might end up. So somebody might not have a really good English, and you want that person to still be able to understand what you did. And that's another reason why adding images and videos to your methods are really good, because images everybody can understand no matter what language they speak. And then just add a lot of notes and recipes, tips and tricks that just would make the reader help to understand your methods, because sometimes you might think, oh, this little detail nobody cares about, but really this might be a little detail that another person might spend like a half a year on trying to figure out why the method doesn't work. So it's really, there's really no limit of what you can add. And with this, I'm at the end of my presentation, and I would like to thank you for joining us today. And if you have any questions, I'm happy to take them now. And you can either type them in the chat window, or I think there's an option for you to unmute yourself. So you can feel free to unmute yourself, and then you can say your question. Thank you.
Thank you, Anita. So there is a question from Neil. I see that you link to the publication, but do you think to the archived dataset associated with the particular protocol?
Hold on, let me. So you can. So that's really up to you. So if you would like to link to your data set, you can, and we encourage people to do so, but that's up to the person who published the protocol to link it to the data set.
Okay. Okay, so Neil said, I ask because about 40% of geo dataset do not have associate publication information. So are to some extent siloed. Having links from protocols IO to such dataset would be one approach to helping close the informatics loop on those datasets.
Right, so you do have the option to link it, but it's really up to, again, it's up to the author to link it, because we cannot, we don't have the capacity to try to figure out which data set goes to which protocol, and like we don't link it, but you definitely do have an option to link it to a data set yourself.
And there is one from Anita Bendrovsky, said reciprocal links between published dataset and the protocols would be really useful, especially for the big data gathering project like Spark and the BICCN.
Yeah, I totally agree. So it would be really good if you can encourage people to do that. And there is an option. So if when you're entering a protocol, you have the option to link to the data sets.
So there's another question. So for existing protocol, are there a way not manually edit and just like uploading some Word file and then edit later?
So, so if you have an existing protocol, so yeah, so you can, I can share my screen again, maybe with you. Let's see. So if you're creating a new protocol, one thing you can do if you don't have the time to put in your protocol right now and you just want to get it out there and you have a PDF or Word document or whatever it might be, is you can put your title and really just put the basic information such as the abstract and like the authors and stuff. And then there's an option to upload a file. So here you would just come and you would just attach your protocol as it currently is. So if that's a PDF or Word document or wherever you have it, you can just upload it here, and then you can publish that version. And then later, if you'd like, you can always come in and create a new version that actually has the steps. But there's definitely an option to just get your method out there with just the PDF or the Word document if you don't have time to enter your protocol right now yet.
Let's see. I didn't see any other questions from the chat box. Yeah, let me go over.
I'm looking too.
Yeah, I think that's all the questions for today.
Okay, and then if anybody has any questions later, please feel free to send me an email at anita@protocols.io, and I'm happy to also answer any questions after this webinar.
Okay, so thanks for Anita for the great talks today, and thanks for everyone joining today's webinar. The recording, like I mentioned before, it should be available next week on dkNET YouTube channel. We'd like to encourage you to share today's webinar with your colleagues and students. To receive upcoming webinar information, please subscribe to our newsletter or check announcement at dkNET.org. Thank you all again and have a great weekend.
Thank you.
Thank you.